Hi, my name is Bak. I'm a molecular biologist and geneticist by training in my bachelor's and have specialized in bioinformatics during my professional and academic life. Here, I would like to present my master's research on the mathematical modeling of haplotype evolution using Wright-Fisher simulations and neural network inference. So why modeling is important? Globally, around 5 million deaths are associated with antimicrobial resistance. That number is higher than HIV or malaria deaths, hence identifying antimicrobial strains become crucial. With the advancement of high computation capabilities, using simulation techniques offer a way to model the evolution of these strains, even before they emerge. And once they emerge, neural networks can provide appropriate simulation parameters for data at hand fast and accurately. And finally, the end goal would be delivering personalized prevention and intervention based on the information given by the neural network model. My thesis will focus on the simulations and neural network modeling parts. Let me give you a background on Wright-Fisher simulations. Suppose we have a population of bacteria with different alleles, and each bacterium with that allele represented as different colors, in this case, red and blue. At each generation, an allele is randomly selected to the next generation. There is no selection, fitness, evolutionary pressure acting on these alleles here, only random selection of each allele. Hence, the frequency of alleles change over time, and sometimes an allele may dominate, or it may disappear from the population. This is a common model to study genetic drift. What we can do with this simulation model is we can incorporate mutations. This time, at each generation, mutations are introduced by a single nucleotide change due to a chance indicated by the mutation rate parameter of the simulation. Again, every allele is selected due to chance, with no evolutionary pressure or fitness. And by the end of the simulation of this model, it gives summary statistics of sequence and allele frequency. And on top of that, by keeping track of the descendants, we can build up a phylogenetic tree, which gives additional tree statistics. Then the question becomes, how to make our simulations more resembling the nature of evolution? An answer to that is by optimizing the parameters that we have used in the simulation based on the true data. For this optimization problem, I have used a neural network due to its ability to learn complex patterns and applicability to large datasets. Here, you see the neural network structure that I have used, which was trained on the simulated tree, allele frequency, and sequence statistics in order to predict for population size and mutation rate parameters. And here are the results that are parameter predictions made on the simulated synthetic test data. The R-squared values for both of the parameters lay around 0.9, hence the majority of the predictions fall near the identity line, thus estimating the true values, while some predictions are overestimate or underestimate the true values. As a result, for the effective population size, underestimated values and overestimated values, and this is for the mutation rate parameter inference. Of course, in order to validate the model's true success, it needs a real-world case data. So I have used Richard Minsky's long-term E. coli evolution experiment data, which has over 75,000 generations of 12 E. coli populations. The left-hand side graph illustrates predictions for mutation rate of 4 colonies that were comparable, while others were significantly overestimated. This may be due to the fact that mutation rate in real world changes throughout the generations, and the different sequence segments may have different mutation rates. So let's continue to the graph on the right, which is in logarithmic scale and showing the result for inferred population size parameter. 3 of the colonies were in acceptable range for population size, while the rest were overestimated. Similar to the mutation rate, this may be due to the population size in real world is dynamic, and the factor of fitness on the colony's reproduction rate may feed to this dynamism. Also, the model's training data was lacking the population size over 1,000 individuals. And finally, if we look at both of the parameters, we can say that my neural network could identify one of the 12 E. coli populations. To conclude, training neural networks with simulation data is quite possible instead of training them with real-world data. And using neural networks for parameter inference instead of other likelihood or likelihood-free methods looks promising. Also, the approach of using Wright-Fisher simulations and neural network inference may be applicable to other research fields, such as cancer evolution in tumor microenvironments. For the future research, we can improve simulations by introducing more complex mutations, such as structural mutations, instead of only thinking of single nucleotide polymorphisms. For the neural networks, we may try different structures, like convolutional neural networks or encoder models. And instead of model predicting for a single value, we may improve it to predict a bounded distribution for parameter inference. And lastly, we can incorporate real genes to study gene evolutions to understand better antimicrobial resistance. And with that, I would like to thank you for your attention. I also want to thank Dr. John Lee's Pathogen Informatics and Modeling Group for accommodating me at the EBI. And lastly, I would like to thank French Embassy in London Scholarship for funding my research. I hope to answer the questions you may have in the live session. For now, bye.